You that the panel discussion will be moderated by Giorgio Trinchieri from NCI. Florencia, you are the first speaker, so I will hand it over to you.
Okay, can you see my first slide?
So we see your slides, but we also see your notes. Did you have presenter mode on?
Let me see, not for the invitation. So I'm going to talk about the role of the gut tumor axis in tumorigenesis with specific focus on pancreatic cancer. So we've been hearing more and more the concept that not only the microbiome lives in the gut, but there's also microbial ecosystems all over the body. So as I said, I'm going to focus on pancreatic cancer, mostly because this is the cancer that I treat. This is the cancer that I focus as an oncologist. It's a very aggressive malignancy, as most of you will already know. With a survival of 9% for 5 years. We think that it's a cancer that really needs new novel strategies for treatment, as really the progress has been very mild. There's, however, one patient in several, usually like 1 in 40 or 50, who actually lives long-term after surgery. And there's not very clear reasons for why is this happening. Many genomic studies have shown that those patients are not different than the others in terms of genomic composition of their tumors. So we've been very curious on trying to understand what other differences exist on those patients, and can we actually understand that better so that we can improve survival on the rest of the patients. So I quickly will jump to the first paper, which actually showed that the microbiome plays a role in pancreatic cancer. So George Miller from NYU has shown a couple of years ago that if you take a mouse, a KPC mouse, which is the standard or the gold standard pancreatic cancer mouse model, spontaneous mouse model, and you put it in germ-free condition, what happened is, as you can see here, there's a delay in tumorigenesis compared with a control mouse, germ mouse, in regular conditions. So there's very clear differences histologically. Now, if you rescue the model by giving by oral gavage feces from a wild-type mouse, there's slight increase in some of the dysplasia, but there's clear rescue of the phenotype when you include stools by oral gavage from a mouse that has pancreatic cancer. So suggesting that you do need bacteria for the pancreatic tumors to start, and then clearly proving that with the oral gavage transplantation system. So further along, another group from Florida has, around the same time, showed in a second type of model, in a subcutaneous model, that treatment with antibiotics can delay pancreatic cancer growth using a subcutaneous KPC cells coming from the same mouse model that I showed before, implanted onto syngeneic mice. And then if the same thing happens, if the same thing treat. Hello?
No, Ben, I just made some rice.
Okay, I'll keep going.
Okay, we can hear you.
Okay, so when the same experiment is done in a rank 1 knockout mouse, you can see the effect of antibiotics is lost, suggesting that you do need the immune system for the bacteria to mediate the tumorigenesis. This is the first paper, really great paper from Ravid, from the Weizmann Institute, who has shown that bacteria is present in pancreatic tumors. And it's a really long story how it was found, but not only he has found that there's bacteria, especially gamma protobacteria, which lives in the pancreas here detected by 16S, FISH, and LPS, but also he went to show that bacteria produces an enzyme which is really important for the activation of chemotherapy, and perhaps that could be one of the reasons that explain why chemotherapies have very limited efficacy in pancreatic cancer. So the question we asked was, does human pancreatic intratumoral bacteria affect outcomes independent of chemotherapy or immunotherapy treatments? And to answer that question, we really don't have responders, clear responders and non-responders to this aggressive cancer. But as I mentioned before, we do have those few patients who respond after resection, where what I mean is they don't have a recurrence after resection, and they go to live long term, more than 5 years. So what we decided to do is we designed 2 cohorts. The first one, the discovery cohort at MD Anderson, and the second discovery validation cohort at Hopkins, in which basically patients were matched from short-term and long-term. As I said, the long-term survivors are the patients that live more than 5 years at MD Anderson, more than 10 years at Hopkins, and the short-term survivors were matched for most of characteristics that we could control. And then specifically, the MD Anderson cohort had neoadjuvant therapy, but most of the Hopkins patients did not have. So what we did was 16S sequencing, and we also did immunoprofiling for those tumors, and we then did the typical analysis of reconstruction of bacterial phylogenies. So the first finding was that the patients from the long-term survivors cohort from both groups were found to have higher microbial alpha diversity, which certainly was interesting, and we hear a lot about the correlation between gut microbial alpha diversity elevated and correlating with better outcomes, but this was related to the tumor microbiome, which was kind of unique. When we went to look at beta diversity so that we could look at differences between the groups, we did find that there was a clustering based on beta diversity in both groups. And farther along, when we put the 4 groups together, short-term and long-term from both cohorts, we could see that, you can see here with the turquoise and the orange, that the long-term survivors from both cohorts were kind of clustering together. When we looked at species, even, at genus level, as I show in this picture, but even later deeper at species level as much as we could, we did find that there's 3 genuses shown here, which really represent less than 5% of the total composition of the patient's tumor microbiome. But nevertheless, they were present on most of the long-term survivors and absent in most of the short-term survivors, as you can see here. And if you put them together with Bacillus clausii, which is the most common species found on the long-term survivors, we were able to get AUCs predicting long-term survivorship really high on the group of MD Anderson and then on the validating cohort of Hopkins. So again, suggesting potentially a predictive role of this signature, which of course will have to be validated in prospective studies. So the next question we had was, is this just an association? Can we use this information to sort of understand if we can change the microbiome? Can that change any outcome? So to understand that, we need to understand where the bacteria are coming from, and for that we performed this study in which patients that were going for a Whipple surgery, they had their tumors resected, and the resection was dividing the tumor, and then we also got specimens that were including adjacent, which is not really normal, but basically non-malignant surrounding tissue, and then stools collected the day before from the same patients. And then those 3 samples were analyzed, and then this is the type of graph that we built when we did 16S sequencing, basically based on the origin of the bacteria found on each of those 3 specimens. We were able to find that in the tumors, around 20-25% of the bacteria was coming from the gut, as you can see, whereas there's like another 25% which is also present in the normal, and then there's like another 50% that it's not in the normal and it's not in the gut, which we think very likely it's coming from the upper GI areas, as other recent studies have also shown, looking at cyst and IPMN and duodenal fluid. So with that information, we asked, can we modulate pancreatic tumors by changing the gut microbiome? So to do that, what we did is basically built a microbial humanized mouse. For that, we use specimens coming from 3 types of patients: short-term survivors, which are basically patients that have been recently diagnosed with pancreatic cancer, all of them metastatic, patients that are long-term survivors. They have had cancer more than 5 years ago, but they have now no evidence of disease. We do not have stools from the same patients that were in the study because, of course, we did not collect stools 20 years ago from these patients. And the third group is basically healthy controls. And what we did is put them onto mice. And again, at this point, we were not expecting necessarily that we are going to find the same bacteria we found in the previous study because, as you know, that was in the tumor, and here we are using stools from these patients. But nevertheless, based on the idea that PDAC creates a dysbiotic gut, which actually kind of feedbacks and supports tumorigenesis, we thought, can we change that? And if we change the bacterial composition with a healthy control of the long-term survivor, can we actually improve outcomes on a mouse? So using the orthotopic mouse model, we asked first the question, can we stably keep differential gut colonization with different donors? And as I mentioned, this is the model of orthotopic implantation of KPC cells on a syngeneic mouse that have been treated with antibiotics and then have been transplanted with feces from these 3 types of patients, and then at 35 days they were sacrificed. And this is what you can see when you analyze by 16S the stools from the 3 types of patients, the 3 types of mice, transplanted with the 3 types of patients. They are very clustered really differentially. The next question was, can the gut microbiome modulate not only the gut but also the tumor microbiome, which is basically the initial part of the talk. And indeed, we found that there were clear differences at the tumor when we did transplantation with different type of donors, as you can see here. So yes, the mice that received FMT from the different type of donors had differential modulation of the tumor microbiome. The next question was, can this microbiome modulation affect tumor immune activation status? When we looked at the immunoprofile of the tumors on those mice, we found that the tumors from mice that had received the long-term survivors' stools had the highest number of activated T cells, the lowest number of suppressive cells. On the contrary, mice that received the stools from the patients with pancreatic cancer had the lowest number of activated T cells and higher number of immune suppressive cells. And finally, we asked, basically, can we change the tumor size by doing this type of transplantation? Our hypothesis was that the short-term survivors were going to have the largest tumors, and then we were not sure if there was going to be a difference between the long-term survivors and the healthy controls, as both of them do not have tumors currently. And what we found was actually both things were happening. So indeed, our short-term survivors' stools transplanted into mice generated the tumors of the largest size, but also we found unexpectedly that the long-term survivors with no evidence of disease patients that did have cancer in the past had the smallest tumors with respect to the ones transplanted with healthy controls. So the final question was, is this effect, especially the long-term survivors' anti-tumoral effect mediated by T cells? We did neutralization with CD8 monoclonal antibodies at the end of the treatment, and what we can see is that you lose that protective effect given by the long-term survivors' transplantations, basically concluding that yes, T cells are mediating this effect. So the conclusions are, number one, human PDAC tumor microbiome diversity and immune activation status correlates with survival. Number two, the PDAC microbial signature of long-term survivorship that we found that, as I mentioned, has to be still validated prospectively in studies. And number three, there's a gut PDAC tumor microbial axis which can modulate tumors' immune activation status and growth. What's next? First, we are very interested about the potential of biomarkers for risk stratification, early diagnosis. We are very interested about the potential preventive role of understanding specific microbes of community, specifically pathogenic role of some of those communities, and understand how we can reverse that. And finally, in terms of therapeutics, we are very interested in the role of FMT in combination with therapies for pancreatic cancer patients. And finally, as William Russell, who was one of the first scientists to see bacteria on tumors, he says it seems almost needless to add that there remains abundant work to be done in this important and attractive field. Thank you very much. And this is just a picture of my lab, and the top, the people that led the work, Jennifer Wargo and Ivan Maitra, Cindy Sears, Drew Pardols, and others who have been great collaborators. Thank you very much.
Wonderful. Thank you, Florencia. Gal, you're up next.
Elizabeth, can you see the slides?
Yes, we can see you and we can hear you too.
Okay, thank you. I was not sure. So first of all, please, I would like to really congratulate the NIH-AACR team for successfully switching from an on-site conference to a completely virtual in really no time. So really, congratulations for that. And I would like also to thank you for the kind invitation for me to present our work on the treatment of refractory melanoma patients with FMT and immunotherapy. So here are my disclosures. Over the past few years, starting with the pioneering work from Lawrence Seefogel's group, we've observed the surge of papers from the strongest groups in the world, indicating that there is a strong association between the composition of the microbiome and successful immunotherapy. And roughly at the same time, there have been several publications, such as this one by Naama Geva-Zatorsky, indicating the mechanistic causative relationship between the microbiome and its ability to shape the immune system of the host. So obviously, one of the most interesting questions from the practical perspective is can we reprogram the immune system in patients by altering the microbiome? And in order to answer that, as a melanoma oncologist, our main challenge here today is the treatment of the refractory metastatic patients. And together with my partner, Dr. Ben Bursi, and my excellent PhD student, Erez Baruch, we hypothesized that if indeed the relationship between the microbiome and the immune system are of cause and effect relationship, then alteration of the microbiome should or could lead to an immune response and hypothetically to a clinical response. Since at the time that we conceived the study, and actually even actually to this day, it is highly unclear which bacteria or consortia are the main drivers for the of the immune system within the context of cancer patients. So we couldn't know who would be the donors if we went to the healthy population. So we decided to rely on donor identification based on clinical characteristics. And if indeed this relationship is a cause and effect, then according to Cox postulates, if we would transfer the microbiome, the whole microbiome of patients who achieved durable and complete response on anti-PD-1 into recipients that have progressed on all lines of therapy, something should happen. So our recipients were refractory patients that have progressed on standard of care treatment with confirmed progression on previous anti-PD-1 with 2 consecutive imaging studies. And the idea was to treat them with FMT and reinduction with anti-PD-1. The recipients underwent native microbiota depletion with antibiotics and PEG-based diarrheal solution, followed by an induction FMT with colonoscopy and orally ingested capsules. Then the patient underwent through repeated cycles of maintenance capsulized FMT and anti-PD-1. The patients were evaluated with imaging studies and also at the tissue levels, both in the gut and in the tumor, with biopsies obtained at baseline and at 1 month or 2 months, respectively. The primary endpoints were safety and microbiome alteration. Secondary endpoints were alteration of the immune system, focusing mainly at the tissue level, both at the gut and at the tumor level. And as exploratory endpoint, we monitored for RECIST-based response. out of dozens of potential donors, we ended up with 2 donors who complied with the rigorous safety criteria, both of the FDA and the European guidelines. And as you can see here, when we analyzed the microbiome, they were quite different. And we could identify different taxa that have been previously associated with successful immunotherapy, but the donors were rather different. The patient population of the recipients were a classical phase 1 population, and we had 10 evaluable patients who have all been heavily pretreated, and 8 of them actually had visceral or CNS metastases, high LDH, or high burden of disease. In terms of safety, we observed none of the patients demonstrated the grade 2 or higher FMT or immune-related adverse effects, so it is safe. And interestingly enough, 4 of the patients had grade 3 immune-related toxicity on their previous immunotherapy exposures, and none of them had any immune-related adverse effects on the trial. We monitored for the microbiome composition with deep sequencing 16S, and the patients were treated, 5 patients were treated with FMT from donor 1, 5 patients were treated with FMT from donor 2, and you can see here, according to the color coding, that pre-treatment, the donors or the recipients were all indistinguishable. However, after 2 months, the donor 1 recipients were clearly different from the microbiome perspective from the donor 2 group, indicating that we have been able to alter the microbiome of the recipients. And when we took a deeper look at the taxa level, we were also able to identify different taxa that were statistically significantly evident in one of the donor groups as opposed to the other. And you can see here that there have been several taxa that have been isolated only or identified only in one of the donor subgroups. Now, moving forward to the alterations of the immune system. The biopsies have been evaluated by immunohistochemistry for CD3, 4, 8, and CD68. And the evaluation was done by 2 expert pathologists, which have been blinded to essentially any parameter, as well as with digital pathology. This is an example of the gut staining for CD68. It is very easy to identify the robust increase in the CD68 staining expression and count in this particular example. And as a matter of fact, we have observed this sort of observation in 9 out of the 10 patients, demonstrating a robust increase in CD68. This is an example of now let's take a look at the tumor biopsies and looking at the most interesting parameter, that's CD8. Over here on the left, this is a tumor with an immunological desert. This is an immune desert, very poor infiltration of CD8 plus T cells. And here on the right, it really became a hot T cell inflamed tumor. And we observed this T cell inflammation in 6 out of the 10 patients. We also tested not only for T cell infiltration, but also we conducted RNA sequencing experiments on the biopsies. And this is just one of the analyses in which we look for immune signatures which are associated with or are indicative of cytotoxicity, effector functions, APCs, exhaustion, etc. And over here, it was very interesting to note that only among donor 1 recipients, we were able to identify the increase in this gene signature, which is indicative of immune activity and function. We could not observe something similar in donor number 2. So there is a difference between infiltration and immune function. Among the gut, we observed only a significant increase in the CD68. And in terms of the tumor, the CD8 increase was almost significant. Now, moving to the last part, and that's the exploratory analysis of response. And here the results were rather striking, because out of 10 treated patients, 3 of the patients exhibited an objective regression of their cancers. 2 patients exhibited a partial response, and 1 patient exhibited a complete response. And interestingly enough, all of these 3 patients were all from the same donor, that's donor number 1, the donor in which we identified the increase in immune signatures in RNA-seq. So clearly differences between the donors. This is the these are the PET-CT scans of the complete responder, and as you can see here, this patient initially exhibited a pseudo progression, and only then he achieved the complete response. And as a matter of fact, this patient did yesterday or 2 days ago his most recent PET-CT, and he's still in complete response, and that's 20 months already into the trial. So to summarize, we observed no safety issues. In terms of the microbiome alteration with FMT, it seems to be feasible, but there are differences between the donors. In terms of immune alteration, we observed immune alteration both at the gut level and at the tumor level in different markers across donors and recipients. However, it is not always, it doesn't always translate into immune gene signature activation and also to clinical response. And in our trial, all clinical responders came from the same donor. And that leads us to multiple open questions. Then these are just exemplar questions. For example, protocol related, should we use colonoscopy or oral capsules or both? Is the eradication of the native microbiome really needed? Is repeated dosing needed? Why are there are differences between the donors? What is the role for dietary modifications? And how should we select in the future the patients or the donors? Is it going to be patient-based or healthy donor-based? These are all questions that needs to be addressed. Our next steps are going to be to move to the second part of the trial, or to the next trial, which is going to be with 41 patients who are going to beef up the data with additional metastatic melanoma patients, and also include new indication of non-small cell lung cancer patients. We're going to utilize 5 new donors, and we essentially have the approval already waiting, but we will be able to commence with the trial once the COVID-19 episode will be over. Essentially, we call this approach is to create evidence through, it's a medicine-based evidence approach, and we think that by utilizing this causative data, we may have a better chance to identify which bacteria or genetic features or immunologic features are really important in this interaction. A lot of people have been involved in this trial. I would like to thank particularly and acknowledge Dr. Erez Baruch, who has completed his PhD based on this trial, Ben Bursi, my close partner, Osnat Mazuz and Adaz Givon for coordinating the trial, Irs Barshak for pathology, Adila at gastroenterology, Omri Koren and Nama Bloch for microbiome analytics, and Ilan Jungster for preparation of the fecal capsules and for intellectual contribution. Thank you.
Thank you so much, Gal. Burkhard, you're up next.
I wonder if Burkhard is aware that the sound isn't coming through. You might have to unmute yourself.
Yes, we can hear you now.
Yes.
Thank you.
Wonderful. Thank you very much. So I will talk about. Do you see this on the screen, or you just see my full screen?
We see the
perfect.
slide.
Don't worry.
Yes.
Very good. Excellent. Yeah. So thank you very much for the invitation and the possibility to participate in this exciting conference. The title of my talk is about microbiome and myocarditis, or in other words, when it comes to matters of the heart, don't always trust your gut. And I will in essence, start with my disclosures, which is just a suggestion that it might be possible to use antibiotics to treat myocarditis. We have heard during this talk, during this meeting, that myocarditis is a prominent side effect of checkpoint inhibitor treatment, and myocarditis is not a so rare condition in humans. 20 to 30 individuals in 100,000 can contract myocarditis, and the pathology is outlined here briefly. What is thought that leads to myocarditis, and the final problem, the severe disease, which is dilated cardiomyopathy due to excessive inflammation in the heart. So it starts, at least that's the theory, with infection with some pathogens, the myocarditis ensues, and the end stage is the inflammatory cardiomyopathy. We have been working on the importance of different T helper cell subsets and thymic negative selection. What is important to mention, also to relate what we heard this morning, is that thymic selection plays some role in this myocarditis, at least in the conditions that we're studying. The data that I will show have been published last year. This is the paper, and the essence is that, to summarize the paper, to picture it, that there are cross-reactive epitopes derived from Bacteroides species that can activate in a cross-reactive fashion truly heart-specific CD4 positive T cells, and that then are conditioned to react with to the production of interleukin-17, so they are Th17 cells. They can migrate into the heart. They receive a different instruction program. They're conditioned to produce Th1 cytokines, including interferon gamma, and together this interaction makes and drives the disease, at least in a mouse model. So which mouse model is suitable to study that? And this is a mouse model that we have generated, and that mouse model comprises of a transgenic T cell receptor that recognizes a truly heart-specific antigen, and this is the myosin heavy chain 6, which is the most abundant protein in the heart. And an important information relating to immune tolerance is that this antigen is in the heart, but it is not expressed in the thymus. So T cells that in a mouse that is called TCRM, so T cell receptor transgenic for myosin T cell receptor, express two different T cell receptor chains, and in comparison to control mice, these transgenic T cell receptor transgenic cells are highly abundant in the transgenic model. So what happens is almost all CD4 positive T cells are specific for one antigen in the heart. This is shown here, that there is these mice develop spontaneous myocarditis, and at the age of 6 weeks, all mice have developed myocarditis, and they progress to inflammatory cardiomyopathy. And the inflammatory cardiomyopathy looks like that. At the age of 8 to 12 weeks, up to 16 weeks, the hearts of those mice that are infected are big compared to the wild-type mice. The left ventricle is dilated, and the wall of the left ventricle is sometimes of the right ventricle, but mainly the left ventricle is damaged. And the damage is due to excessive fibrosis, as shown here. And the immunological mechanism has been published. So the microbiome plays a role in this mouse model, and this is seen here because the mice, not all of the mice, develop dilated cardiomyopathy. So the transgenic mice develop cardiomyopathy. If they are raised under germ-free conditions, none of the mice succumb to inflammatory cardiomyopathy. This is seen also here. Under specific pathogen-free conditions, the mice develop a severe disease with excessive fibrotic damage in the heart. If they're raised under germ-free conditions, they're free of this pathological condition. We can also monitor the clinical appearance. So mice that are raised, or the T-cell receptor transgenic mice that are raised under germ-free conditions, have normal heart function, for example, here shown as ejection fraction measured by echocardiography. The disease can be transferred if germ-free mice receive or are exposed to a normal microbiome from in cages by littermates, so they are co-housed at the age of 6 or 4 or 8 weeks. Immediately after the exposure to the microbiome, they develop the disease, and the progression of disease follows like in the SPF, so specific pathogen-free. So what are the mechanisms? I'm cutting the story short. A very pathogenic cytokine that is involved in the cardiac damage is interleukin-17. The take-home message is here: if there is no microbiome, there's no IL-17. Under co-housing conditions, the IL-17 is back in the CD4 T cells that infiltrate the heart. How does it happen? Where are these heart-specific T cells activated? The activation occurs in the colonic lamina propria. This is a histological representation of the colonic lamina propria with a colonic patch, and the green cells here are T-cell receptor transgenic cells specific for the heart. They are activated early on in the colonic lamina propria and in the mesenteric lymph node. Only after they have proliferated later they can go into the heart. All these data suggested that there is cross-reactivity, that the heart-specific CD4 positive T cells are activated in the lamina propria, and indeed some differences in the microbiome are present, mainly in the Bacteroides species. So they are more abundant in transgene negative, so in normal bulb C mice, but whenever a T-cell receptor specific for the heart is present, these species were reduced. The cross-reactivity could be pointed to a specific peptide. What we see here is the myosin heavy chain 6 peptide that is immunogenic in bulb C mice with a very efficient binding to the respective MHC class 2 molecule, and there is similarity to the human peptide, and 2 Bacteroides strains have been identified, Bacteroides species and Bacteroides theta iota omicron, that share these epitopes that have a very high predicted binding efficacy. This is a bit immunological nerdiness, but just to show the data and to communicate, these are truly cross-reactive CD4 positive T cells, specific for the heart, but reacting to the bacteria, specific for the heart with a high binding efficacy, but sufficiently cross-reacting with the beta-galactosidase epitope from colonic commensal. So if there is a pathogenic principle in the gut, the question was, can we treat? And we use different combinations and different antibiotics. And this is possible in the fecal DNA to treat and to reduce the abundance of Bacteroides theta iota omicron DNA, and that is associated with a significant and substantial reduction in the disease severity. So this disease severity degree 2 reflects a very mild inflammation in the heart. Can that be translated to human myocarditis? We tested first, and this is only the highlights of some of the these are some of the data that we have published. The Amitis cohort is a biopsy-proven myocarditis cohort, and this cohort has been followed. This is the serum reactivity against Bacteroides theta, immunoglobulin IgG. What can be seen is that there's a significantly increase of the seroreactivity against Bacteroides theta at diagnosis compared to healthy controls, and this reactivity decreases. In this cohort, the reactivity is specific for Bacteroides theta iota omicron. Other Bacteroides strains in the healthy and in the myocarditis patients are not different. as well as other bacteria that we have tested, Enterobacter cloacae or Escherichia coli. The differences in the seroactivity is not given. Not only is there a specific reactivity against Bacteroides theta, there's also specific cross-reactivity between the B theta peptide and the myosin 6 peptide in human T cells. Predicted binding to a specific set of HLA alleles can be observed here, so there is cross-reactivity predicted, and we can demonstrate that in patients. So in our cohort, in the healthy cohort, HLA DQB103 alleles, these are these here, for example, they have the right HLA that could bind both peptides, but they don't have disease. They have low or no reactivity. Myocarditis patients with HLA alleles, so non-DQB103, so for example those here, they have no or low reactivity. Myocarditis patients with potential MHC alleles, class 2 alleles that bind both peptides show significant and very high reactivity, T cell reactivity here, read out as interferon gamma production in the peripheral blood. So that leads me to the summary of these findings. Microbiota should be or contain, most likely in human myocarditis patients, a particular species with a bacteria beta-galactosidase that is cross-reactive to the human myosin 6 epitope, that can bind to a particular set of HLA alleles, here HLA DQB103. What is missing, not all of those patients that have the bacteria and that genetic background automatically develop disease. It appears, and this is the conclusion that we have from our study, that other cardiac damage and or damage to the gastrointestinal tract is necessary to trigger, to kick off the disease so that the individuals in the end, so that the patients develop the disease. What is the further impact and the outlook? In checkpoint inhibitor treated patients, we've heard that myocarditis is the most lethal side effect. And for example, here in the 2016 New England Journal of Medicine study, what has been found was that patients that suffer from myocarditis have exactly or possess HLA DQB10301 alleles, and these are the alleles that bind most likely both the Bacteroides theta, iota omicron, beta-galactosidase, and the human myosin 6 epitope. What could then be the impact for cancer patients if, for example, such cross-reactivity between self-antigens and tumor antigens is given, and the cross-reactivity is so self-antigens, tumor antigens, and bacteria antigens, then antibiotic treatment should have an impact on the efficacy of the checkpoint inhibitor treatment. We would like now to test in a larger trial, in a controlled trial, to manipulate the gut microbiome first by repurposing an approved antibiotic, non-systemic broadband antibiotic, and this is rifaximin, to treat myocarditis patients, and to further elaborate whether and to what extent presence of particular bacteria in the gut predispose to the development of myocarditis. Coming to the end, so these are all people who have contributed to the study, my collaborators here in St. Gallen, people in Calgary and Bern, and others who have contributed. I would like to thank the Swiss National Science Foundation for funding. Thank you very much.
Wonderful, thank you, Burkhard. So now we'll proceed with the panel discussion. If everyone would like to share their video or their webcams, their video, so we can see you. And Dr. Trinchieri, just a reminder, if you would like the questions from the audience, please just say what are the questions, and we will read those for you. Thank you.
Okay, Thank you, all the three speakers. So we hear about the role of the intratumor microbiota in pancreatic cancer. We discussed the treatment of refractory PD-1 refractory melanoma patients with fecal microbiota transplant, as well as the role of microbiota and antigen mimicry in cardiomyopathy. And if so, if there are questions from the panel, I would like to see them from the audience. But maybe I can start with the first question from Florencia. And I would like to know whether you've been able to evaluate where the microbes are in the tumor, particularly are they viable, can be cultural to the tumor, are they intracellular, if they're intracellular, are they in a tumor cell or in inoculated? Florencia?
So we certainly have done that. We tried to do it with the long-term survivors frozen material we had. Unfortunately, as you well know, you will have to do several type of conditions for different type of cultures to grow different type of bacteria. So we basically tried to go for the ones that were more frequent, and we were able to grow some of them, like Saccharopolyspora and others. But the group from Ravid, he has been, Ravid Strassman in Israel has been really looking at this, and in their publication they actually postulate that the bacteria is intracellular, is present in the tumor cells, and the idea will be that when the tumor cells uptake gemcitabine, some of the alteration metabolism has to do with the direct presence of the bacteria in the cytoplasm, and liberation of enzymes that can affect the metabolites of gem. But we are now further looking at that, because we are also interested on different type of immune cells that we have also found that can have the bacteria as it's expected, especially antigen presenting cells. So we are exploring now kind of in a kind of broader manner. But yeah, so far the published data suggests that it is intracellular.
Thank you, Florencia. So Gal, I was just curious. I know you've been following the composition microbiome for a long time after the fecal transplant. Is any case in which the microbiome revert back to the original patient microbiome or remains stable for quite a long time?
So this is obviously a very important question with deep implications. At the moment, we have the analysis in place for up to 2 months. We're still working on the analysis of the longer follow-ups in order to understand how stable the microbiome is. And as a matter of fact, this question is very strongly interlinked with the question, with one of the open questions that I raised. For example, the necessity of recurrent or repeated dosing. Is it really necessary or not? And even in the case of reversion back to the original microbiome, it would be important to note whether it is associated with clinical failure or stability of the success. So I hope that over the upcoming months we will have a clearer answer for that, at least based on our small sample size.
Okay, thank you. And Burkhard, you've been showing quite dramatic data with Bacteroides Omicron and the cross-reactivity with beta-galactosidase, but you also showed the other beta-galactosidase or other proteins from other bacteria can also show cross-reactivity with the autoantigen, with self-antigen. Being able to do any notobiotic microexperiment, just adding some of these one by one and see if they can affect their response?
We did some recolonization, so single recolonization with Bacteroides theta iota Omicron alone and with B theta lacking the beta galactosidase. So without the beta-galactosidase in the recolonized T cell receptor transgenic mice, the activation of both the anti-Bacteroides theta response and the response against the hard antigen was reduced, which is, of course, it's an antigen. It's a further evidence that it's a truly antigen cross-reactivity, that that bacteria can introduce mimic peptides that are sufficient to activate self-reactive CD4 T cells. So if under conditions under checkpoint inhibitor treatment, such cross-reactive T cells would be unleashed, it would be one idea that patients could then profit from such cross-reactivity against the microbiome.
So do you say that you're being able to do that in monoclonalization? So the bacteroides by itself, if the antigen is present, is sufficient to induce an immune response in the host?
Yes. There is some general inflammation, of course. That's how the immune system works. The Bacteroides theta without the antigen leads to some immune activation, and under conditions of excessive presence of antigen-specific T cells, there is a basic activation. But antigen specificity, at least under these conditions, when an antigen is restricted to a target organ and is not present in the thymus, then the effect can be focused on the target organ and maybe even on a tumor, if there would be cross-reactivity. And
just as curiosity, were these experiments done in germ-free mice or in antibiotic-treated mice?
Both in germ-free and in antibiotic.
Thank you very much. So we have several questions now. So one question to stand myocarditis, the question whether there is enough patients to reach a sample size sufficient to indicate to study whether microbial intervention can mitigate myocarditis.
So
that's the feasibility of the study question.
Yeah. We have planned such a study, and if we use cardiac MRI, which has a very good resolution, and there are previous studies that demonstrate that quite exact measurement of the cardiac inflammation is possible. So we would end up with a sample size of 150 patients that need to be overall recruited to the end of the study, split by 2. It's roughly 75 per group, treated versus untreated.
In the FMT study, one question has been asking is whether in the case of metastatic melanoma, would it matter the location of metastatic tumor of the recipient for the transplant to be successful?
So we, at least again, taking into account that the sample size is limited, it doesn't seem to correlate with location of the metastasis. It is a possibility that, as in all or almost all treatment modalities, that there will be a relationship with the disease burden or the kinetics of the disease, but this remains to be tested or investigated in the future studies. This is one of the reasons for our objective to expand the cohorts in order to learn more about the parameters that are associated with response or failure of the treatment.
Yeah, the next question is here from Sarah Monte from Toronto. It's similar, and I think it's both for Gal and Florencia. Is the gut leaky because of the host as a tumor? And is the microbiota from gut entering the tumor? I think that's applied both for a tumor microbiota and fecal transplant. So if Gal and Florencia like to address this question.
Yeah, that is a great question. So I mean, there is a possibility. We are looking now. We're using mouse models in which we can track permeability and kind of use that as a variable to see if we can change, using the type of transplant, can you alter the permeability and, then observe what is the difference in sort of tumor bacteria colonization. That is one variable. The second variable, the fact that we can get different bacteria into the tumor than in the adjacent area suggests that there's also some type of, local sort of permeability that has to do with perhaps, the vascularization or the lack of vascularization of the tumors. So, yeah, I think those are, wonderful, next avenues that we should explore.
I would like to second Florencia's opinion. I think that these are indeed great questions, especially coming into when coming to think about metastatic patients with, for example, with increased inflammatory cytokines such as TNF, which may be, for example, affecting the gut leakiness. And accordingly, the effect of transplanted microbiota, I mean, it would be extremely interesting to see whether the same bacteria could be located in different places. I think that in patients, these sort of studies is going to be slightly challenging, but probably in the murine models, it would be easier to see whether such a relationship exists.
Okay, thank you.
Yeah.
So the next question, yes? Okay. The next question is from our young NCI, again, I think to be addressed both by Florencia and Gal, is whether have you looked at the host metabolome in your study?
Yeah, so we didn't look. So what we did is we used Picross analysis to look at predictors, prediction of metabolites, and we did find some specific pathways that were, just predicted by the bacteria present on the tumors of the long-term survivors versus the short-term survivors. And then what we did was, metabolomics in the tumors from the FMT experiment, looking at the tumors of the mice transplanted, and we did find specifically in, biliary acid pathways, metabolites that were increased on the mice that received long-term survivors transplant. However, I have to say that was done at the end of the experiment. So as as I showed today, the tumor size was different. So we will not know if the metabolomics difference were because of the change in the size and the immunoactivation, or solely because of the transplantation. So we are now doing a second experiment in which we are solely looking at metabolomic changes following the transplantation, even prior to the tumor implantation, and then following up at the end and see if there are differences, and we can get better answers.
In our experiment so far, we haven't conducted metabolomics. This is part of the future plans, but trying to do some reverse engineering hypothesis generation by, for example, by Picross analysis. We haven't been really able to identify major metabolomic or metabolic pathways between the responders and the non-responders among the transplanted patients, but this is obviously just indirect evidence.
Okay, another question from Yaruska Naido at Johns Hopkins, I think can be addressed by all the speakers, is what the relationship between circulating microbial DNA and the gut microbiota versus intratumor microbiota. I think this is a very timely question. There's a lot of interest. A recent paper on the circulating microbial DNA. So, if any of you would like to address this question?
From my perspective, I mean, we are looking into that, but we do not have yet answers. The recent publication in Nature from Rob Knight certainly, shows the power of the serum circulating microbial signature. But we have, we want to do it in a kind of paired fashion, looking at tumor versus gut versus the circulating DNA within the same patient to really, get final answers.
Burkhard, do you think that the circulating DNA or microbial product may have any role in the immune response to the beta-galactosidase, or does something happen at the level?
Whether they have I think they could reflect to some extent, and of course it also depends on the sensitivity of the detection. It could serve as an indicator for the presence of such bacteria. Whether the DNA that really circulates has an immunological effect, probably not. But as a measure, as a means for detection of the presence of particular bacteria, that's certainly possible.
Except the circulating microbial DNA can actually be carried in vesicle or other particles. May also carry the antigen, and the presence of DNA may work as an adjuvant.
Yeah.
The question for Florencia from San Kim is whether, in addition to CD8 T cell, you look at the CD4, NK, and B cell.
So we did not look at B cells or NKs. I mean, we look at Th1. I mean, mostly there were kind of some gamma differences, but there were the biggest difference were in the CD8s and MDSCs, but not really at other tumor types.
Still another question, anonymous question. I think that's mostly for Florencia and Gal, is whether we need a control of the patient diet following the FMT. Florencia, I know that Anderson is doing a lot of that, and Gal, do you have any data about diet?
Okay, so in our trial, we did control. I mean, the patients had to go through a certain diet, because we wanted to try and at least from that perspective, to minimize the variability between the patients. So patients essentially were instructed to follow a Mediterranean diet, and they were followed up with for adherence by clinical nutritionists. But I do think that, especially in light of the different evidence coming from the MD Anderson group about the importance of diet and its relationship with the microbiome, I do believe that this is certainly an extremely interesting field to study, and I do believe that FMT or other microbiome manipulations may require different sorts of diets when trying to manipulate it for different sorts of indications. So cancer may be very different from IBD or from myocarditis or other, or even skin autoimmunity. I mean, all of these may be very, very different.
Florencia, is anything to add?
Not in particular. I mean, I don't think we yet know because we don't have like prospective data looking at specific diet, specific antibiotics, but it is very complex, and I don't think we can assure at this point what antibiotics specifically to avoid, or which diet will be the optimal. But these are things that we have to look, and that has to be explored in patients in prospective trials. So it's just a lot to do.
There's another question about fecal microbiota transplantations. That is one part of the question we have addressed already, whether the transplant microbiome survived. But the second part of the question is whether the immune system mount a response against the microbiome that may include a transplant microbiome may include microspecies to which the individual may have been exposed early in life. And if I could paraphrase the question a little bit to Gal, is that one possibility that when we do the fecal transplant is that we really use a sort of inflammatory response, because we expose the individual to bugs that they are not normally present in their own microbiome. We know in the germ-free mice that is a major phenomenon. You get a major inflammatory response. Have you seen any anything like that in the patient that could justify the effect of the transplant on the immune.
Yeah, no, I mean, from my perspective, I mean, this is actually a very good point. I mean, I would say we will have to experimentally address it. You know, you could think of preclinical experiments with FMT, which is kind of transplanted several times, and then see if you kind of acquire certain like tolerance to it, and then see if the responses you see are kind of different. But also, I think the long-term the long-term colonization of the FMT, I don't think we also know so well, because it's different in healthy patients, it's different on the colitis patients, in which this has been explored a lot more, and in the cancer patients.
I definitely agree with the last part of Florencia's comment regarding whether it mounts whether the FMT mounts an immune response. So I would assume, of course, that it would be the easiest or the most logic place to look at is, of course, the humoral response and generating some data about bacteria-specific or epitope-specific for certain commensals or multiple commensals could be indicative whether such process indeed occurs or not. So I know that there are several groups in the world that are working on that, and I believe that this would definitely be of importance and would be able to provide this question with an answer.
So then we have a couple of questions from the audience of the role of antibiotics, both for anti-human immunity as well as adverse effects. And the question is, which antibody, what is the risk, and which antibody we should use or avoid? And I think that probably this can be addressed by all the three speakers. Burkhard, do you want to start?
Yeah, I could start. Yeah, thank you. So, of course, giving antibiotics, that is a massive intervention, and I think the case, at least in a more global context, in larger cohorts, is quite convincing that at least in checkpoint inhibitor treated patients, that with antibiotics given, response to the checkpoint inhibitor treatment is less good. Whether and to what extent that really is a pathological principle or a therapeutical principle, if we just flip the coin, based on the cross-reactivity between microbial antigens and tumor antigens, I think we would require further studies and to really elaborate that. For example, also in patients that Gal has studied, patient 1 versus patient 2. What kind of, the feces from patient 1 versus the feces from patient 2? Where is a good response and where is a less good response that you have achieved? I think this analysis, detailed analysis on your study, but also future studies, will complement to a large extent the studies that we have in antibiotic treatment, checkpoint inhibitor treated patients. So I don't think there's one answer yet whether giving antibiotics or specific antibiotics would be good or bad for particular tumors. It is probably for patients, particularly for patients, not really satisfying if we give answers like, yeah, it's complex and we require more studies. But I think in this instance, at least where I am as a researcher in the moment, I think this is where we are, because there is a clear link, but we need more evidence, as Gal has said, to really sharpen these therapeutic decisions. Sorry for the patients for evading that. I think there is, at least for me, and I can also admit I'm only a veterinarian, there's no good or bad answer. But Gal, you're a physician, you should know.
Gal? Gal, we cannot hear you.
So it is really hard to accurately make the conclusion from patients. And now I'm putting again aside the patients from the mice, because in the mice, there is, the data demonstrated by several groups for example, from Lawrence's group, showing how the antibiotics can completely abrogate the effect of checkpoint inhibitors in mice. But in patients, to compare patients who received antibiotics to those who did not receive antibiotics, of course, in such a retrospective manner, it is very hard to deduce any significant or to make any significant statements, because one cannot really know why some patients require the antibiotics. And when a patient comes into the clinic with a pneumonia or a bacterial infection, even though the antibiotics may or may not impair the efficacy of the checkpoint inhibitors, in principle, not to treat a bacterial infection with antibiotics, this is, it could put the patient in much greater risk. So this is why, from the practical perspective, we still do not know, and we tend to go with the clinical instinct. But it is extremely important to understand whether some antibiotics or which bacteria are important, bacteria or consortia are important for response. And frankly, I am not sure that, since antibiotics typically are broad spectrum, even the narrow spectrum are broad from the microbiological perspective, so I'm not really sure that we will be able to discern good antibiotics versus bad antibiotics.
Florencia, do you have any comment?
Nothing else to add. Again, I think there's a lot yet to be known, and I don't think yet, at least I don't think we can say antibiotics, necessarily should be avoided, or yes, that's the way to go. So I think we don't yet understand, and there's a lot of association data between antibiotics use and decreased response to therapies in some tumors, but I don't think that is the final word. I think there's a lot more to be known.
So then we have two questions that are mostly related to the pancreatic cancer and patient and model. It's in a way related. One is how early during the tumor growth bacteria colonize the tissue, and I would add they already present in the pancreas, so they tend to reach the pancreas in a larger number when the tumor is growing. And when you have a CD8 T cell response in the pancreatic cancer, particularly following a fecal microbiome transplant in the animal model, are those CD8 T cell recognize bacterial antigen or tumor antigen?
Yes. So these are all questions that we are exploring now. We very recently published our data, and we are looking certainly at specificity of the T cells, because we do know that the mice that received the FNT from the LTS patients did mount an immune response. So those are the mice that we want to focus to kind of further understand, what are the T cells recognizing, and we are doing TCR sequencing and further studies to answer that question, but we don't have an answer yet.
Another question is the correlation between different gut microbiota and either presence, severity, or persistence of immune-related adverse effects in the patient treated with checkpoint inhibitor. So maybe Burkhard, can you start addressing this question? Besides what you already
we still have to infer from the study that we have been doing, or at least that's what I can do. So if there is a causal relationship, as we have seen in myocarditis in humans, that is similar than for antigens that are present in tumors, so that antigens from the microbiome are recognized by tumor-specific T cells, then a treatment with checkpoint inhibitors unleashes, of course, the cross-reactive T cells, mainly the CD4 T cells, and that would then be beneficial for the patient and help the immune system overall to cope with the tumor.
Any other comment on
That
that?
answer the question somehow.
Darko? Would have to say, yeah. Gal and Florencia, any comment or any relationship between gut microbiota and adverse effects in patients?
So there has been several patients, for example, from Jed Wolchok's group at Memorial Sloan Kettering, and also from the Andy Anderson group, linking the severity or proportion of patients who developed colitis following ICI treatment, associating those patients with certain microbiota compositions versus the others. I think that I mean, these sort of course experiments need to be further understood from the mechanistic perspective. And there has been some experience, for example, as far as I remember, by Mimi Wong in MD Anderson treating or curing persistent colitis, steroid-resistant colitis with FMT from healthy individuals. So it seems that from the GI perspective or GI toxicities, it is that there are strong relationship between the microbiome, but we still need to understand what is the underlying mechanism. And it seems that there may be some cross relationship or correlations between the microbiome taxa that are associated with toxicity with those that are associated with response. But again, we must put these in the context that across continents or across ethnicities, these may not necessarily be the same bacteria.
Thank you.
Yeah.
Okay. Stop here.
Sorry.
One more question. Okay.
No, I just wanted to say that I think Mimi Wang is giving a talk, if I'm not mistaken, tomorrow. So she'll expand on this.
So we have a few seconds left, and I'd like to ask probably the most difficult question for Gal. Is you're planning to expand the study from melanoma to non-small cell cancer, I think?
Well, Switzerland is not a very big country, and myocarditis is not a frequent event, and also we just don't have that many focus centers on with patients with checkpoint inhibitors. I think if that is considered a valuable hypothesis that prevention to some extent of the immune events in checkpoint inhibitor treated patients can be obtained to some extent with manipulating the microbiome, that should be done in the larger US American centers where you have enough patients with a particular side effect, and here the myocarditis. And I think that is something, at least if I was in the US, or even if I could travel to the US in the moment, I would love to speak with everyone who would be interested to follow up such a study. Thank you.
Thank you. So, Gad, the last question from the audience is if you are going to expand your study of fecal microbiome transplant to non-small cell cancer patients, you think the same microbiome that enabled the melanoma response will also benefit in non-small cell lung cancer?
Of course, this is an excellent question. We essentially plan, since we do not really know the answer, we plan to do the transplantations for the non-small cell lung cancer patients from non-small cell lung cancer complete responders. So this is the plan. But if I had to speculate, I would assume that the microbiota. I mean, of course, we cannot know that. But I would assume that that it is about triggering basic fundamental mechanisms which would be tumor agnostic, and the translation or the generation of the T cells and the supporting adaptive immunity mechanism should not really depend on the indication. And this is all of the checkpoint blockade is based on that concept. But I think that it would be relatively easy to test this hypothesis in or to test this in murine models.
Okay, thank you very much all the speakers for the discussion. I think it was quite a good discussion. I think probably the audience learned a lot. Too bad that we cannot talk in person and have an audience in front of us, but I think nice. Thank you very much.
Thank you.
Thank you all.
Thank you all. That was a wonderful discussion. So now we'll transition into our last session.